Welcome to Multibiomics again, and today we'll be starting our first lecture on hands-on practice using Surat package. So till now, I'm hoping that you have downloaded your R latest version of R Studio, and you have also downloaded the all the packages that I have, uploaded those packages in the Google Classroom link in the announcement section, and the code is also provided. You can just copy and paste in your RStudio, and those packages will be downloaded and installed, right? So first, for starting with the hands-on experience, let us go to our Google Classroom. You see, this was the announcement that I was talking about. Here, installation guide.pdf, you just open it and you just follow the instruction. For now, we will go to the classwork tab. In that, the first topic, an introduction to common analytical workflow. In this, we are performing clustering and cell type annotation. Just click over here and you get a brief introduction that this tutorial, okay, fine. This tutorial implements the major component of a standard unsupervised clustering. So we'll be performing clustering over here. Okay, and the workflow will include QCs, quality checks, data filtration, calculation of high variance genes. Okay, we will always select those genes. Uh, those are, highly, not related. So the genes who vary. So we see variance is more, right? They are not more correlated, which will help us in dimensionality reduction. And yes, we will be performing dimensionality reduction. And after the dimensionality reduction, we will be performing the clustering, and this will be the graph-based clustering. If you don't understand this right now, don't worry. As we proceed further during the analysis, you'll get an idea of what we are talking about. And then the identification of cluster markers. I mean, the cell will be separated according to different clusters. So, why are those cell behaving like that? Why are they being separated? It is due to some markers, some genes. Here, the markers are nothing but the expression level of the transcripts, right? So, with the help of those data, we'll be also identifying, or we'll be naming them, we'll be annotating them. Okay. So here's the GitHub code. You just click on this and you'll be landing on my GitHub page where you get this code. This is the first code in this series, and this is like clustering and annotation. So you can just copy and paste. I would ask you to don't just copy and paste the script at once. You can copy and paste it either line by line, or you can copy and paste it in chunks, right? So that you can easily move forward. So you can go to file. Okay. Uh, right. So you can go to file, new file, and you open a new R script, and you can just copy and paste accordingly. So first we'll be loading some libraries. Okay, so this is for data manipulation. This is Surat, and here we'll be performing single cell analysis, and we will be needing this package for, you know, patching up different graphs and visualization, all those things. Okay, so the first step is to load our data set. I have given you the link. You can click on the link just below the GitHub link. There's a dataset link also. You just download it. It will be in the form of a compressed file. You just decompress it, and you can store it accordingly into your uh, folder. So for that, you have to go to session and set up your working directory and choose the directory where you have selected your own, where you have placed your data set, right? So I have placed it in different folder. Just going through my working directory right now. Right. So here you see the working directory has changed. Okay. So the structure is like I have kept it in hands-on under hands-on folder, but you will be having PBMC 3K bar gene, BC and matrices. So you have gene data, you have BC, that's barcode, and then you have got the matrix counts. Okay, in that filter gene BC matrix, and inside HG19 data a folder, you have three files. This is matrix.mts, this is genes.tsv, that is tab separated file, and then here again a tab separated file which gives you the information of barcodes. Okay, so okay, these are the larger files which will not open. I'll take another class where I will be explaining the different file formats. Just for now, you understand that the matrix.mts is a matrix where it is a matrix where you have got genes on a row and you have got different cells on columns. So different columns are the different cells and rows represent the genes. And in each data sample is actually the expression level of that gene in the particular cell. And then we have also got genes.tvs. So genes.tvs is also it's a tabular format where the metadata of the genes are present. And in barcodes.tvs, we have got, see the barcodes, I have opened the barcodes.tvs. So I have already told you in my previous lecture video that barcodes are nothing but the unique um, sequences, and each unique sequences correspond to a different cell type. Okay, and if I open genes.tvs, you see you get two columns. This is the gene id. En represent that it is an ensemble id, and then this is the gene name or gene symbol. Usually minus 10, it means that it has come, the data is from the 10x genomics. Okay, no, sorry, not here, not minus 10, sorry, this one, I'm so sorry, minus 1. So the barcodes minus 1, it represents that it has come from 10x genomics platform. And here this is nothing but this is a single column and this is a separate column. This is the gene ID and this is the gene symbol or the gene name. Okay. So these are the file formats. Now matrix.mts is a larger file, so we are not able to open it right now. So let us load this data. Now I have opened this directory as my current working directory, and with the help of code we'll load this data now. So here we are declaring pbmc.data, okay, as a variable, and in that we are going to read the data. So this is my path to the data set, but you should be giving your path, and you should only be giving the folder which contains all these three data sets. So these three files, right? So these three are inside HG19 folder. So you have to give the path only till this particular part. And all this data. Now, this is the function where you read from 10x. The 10x Genomics is a platform. It is giving us the data set. So for reading such data set from 10x Genomics, this is the function that you are using, read 10x. And the directory path is this one. So when we load this data, right, so I just place my cursor over here. There's no need to select the whole line, just place the cursor over here and select run. So here error in reading could not read function read 10x. Okay. It was not showing this error before. So the issue was, what would be the issue? Just okay. So I have not loaded these libraries. So it's very important to load these libraries. Okay, because Surat was not loaded, and this read 10x function is actually a function of Surat library. That's why it was not working. It was throwing an error. Now, if I run this, you see now it is run. And if you see our environment, global environment, we have a DGC matrix. It is a sparse matrix, right? So, what do you mean by sparse matrix? Now, it is a very huge file. There are n number of genes and n number of cells. And in many data points, in many cells inside that, the value is 0. So most of the biological data is known to be sparse data because in most of the places the value returned is 0. So such data is known as sparse data, right? So this is a sparse matrix. If you open and see this data, we will understand that it is a sparse matrix. We will be looking at it, but since you have loaded a data, but we will be creating a Surat object. So what is a Surat object? Surat object is, you understand, when you create a Surat object, you think that it is a container with various levels. So in this single cell assay, we are going to perform dimensionality reduction, we are going to perform quality checks, we are going to filter, annotate, we are going to perform, clustering. Then we will perform, with the help of graphs, we'll be clustering, right? So all those data will be stored into various levels. Now, this is a single cell RNA experiment where you only have the data of the transcripts or the mRNA molecules. But if we go to other protocols such as Visium and Xenium data, which deals with spatial transcriptomics, you don't only have this, the metadata of what is the expression level, but you also have the spatial coordinate that where each cells are present in the particular tissue sample. You also have another data showing protein expression level. So in one single object, you keep all those information in different layers as different assays. So when we are working in this data set, you only have an RNA assay because you have only transcripts. But in the same similar kind of data sets, you can also have proteomics data, you can have genomics data, you can have metabolomics data, you can have spatial data, you can have so many things for a single sample, and all will be it will be contained in this Surat object, right? So you have just simply loaded the data now, but now that data will be converted into a Surat object using this command, create Surat objects. Okay, and the counts will be, what are the counts? The matrix, right? The matrix is nothing but in all the rows there are genes, and in all the columns there are cells, and the values corresponding to those genes and those cells will be the expression level. So those expression level are nothing but the counts, right? And then we are giving the name to this project like PBMC 3K, and what we are going to specify over here, that we are going to keep only those genes because it's a very huge data. We tried to open it in R, but we couldn't because it was a huge data. So here what it is saying, that keep only those genes which are expressed in at least 3 different cells, right? Because if they're not expressed in more than 3 cells, then they are not relevant. We cannot deduce any relationship between the expression of these type of genes. And also here they are saying is keep those data So those genes or those cells, sorry, here you have to keep all those cells that expresses minimum of 200 genes. Okay, so we are just filtering our data and we are just keeping them in our Surat object. So again, if I repeat, the those genes, this one is those genes that has been expressed in minimum 3 different cells, and these should be those cells that are expressing minimum 200 different genes. Okay, only those points will be taken. Now, so we have loaded this, and I'm just placing the cursor in my 8th line here, right? And then I'm running it. So okay, so this warning you can ignore because you cannot have underscores. So R has replaced those names with dashes. Okay, and they have created a Surat object, right? And then a Surat object is stored under the name PBMC, and you can see here also the PBMC, it's a large Surat object. Okay, and then here we can visualize this. We can run, and you see when we run the PBMC here, it is an object of class Surat. It's a Surat object, and it has 13,714 genes, right? Features are nothing but genes across 2,700 samples, and samples are nothing but cells. Now, there's only one assay. Why one assay? Because we only have details of the RNA, right? If suppose this data also has a count matrix of protein expression, so it would be 2 assays, right? So there are, we can have different types of assays in our data. Okay, so only one layer is present. That means only count matrix is present. In spatial transcriptomics, we also have, another layer that is, not only the counts of expressions of RNA or proteins or metabolites, but also there are the spatial data which gives the coordinates. So different layers will be present, but this is a simple data which has only one layer, right? And moving on to the next part, uh, copying and pasting it from my GitHub repository. Paste it over here. Yeah. So let's examine a few genes in the first 30 cells. So we have loaded the data with this name, pbmc.data, and you have to explore your data first. You have to open it and explore the data. So some genes name, as we know, so CD3D is present, TCL1A is present, MS4A1 is present. I would like you to go and Google them and search and research about them, that these are the biomarkers of what kind of cells. I'm giving you a hint that these are the biomarkers for immune cells. Okay, and these are the peripheral. PBMC is actually peripheral blood mononuclear cells, right? So here, so these 3 genes will be looking in the first 30 cells. It's not hard to understand these codes. If I run this, you see now, so 3 into the matrix of 3 into 30 dimensions. So it is having 3 rows and 30 columns. Why 3 rows? Because there are 3 genes that we have selected from our whole data. We are just subsetting our data, right? And then we have taken the first 30 cells. So that is why there are 30 columns. If you see here below in the console, you can see there are so many dots. Okay. So what do you mean by dots? That means the value here is 0. So if you have more number of 0 that are present, okay, that means it is a sparse matrix. And this is a common problem when it comes to biological data. Now, what do you mean by these numbers? So the first one is the cell 1, the first cell. And here the expression level of CD3D is 4. So it is how you can say it is 4. That means 4 UMI are 4 different types of. I'm not saying that there are only 4 mRNA molecules present. There may be thousands and millions of mRNA may be present, but there are 4 different types of mRNA which has 4 unique UMI, unique molecular identifier. So these are the expression levels that we are getting. Okay. Similarly, for TCL1A gene, and this is for MS4A1 gene. So wherever there are dots, that means the value is 0 and they are not being expressed in that cell, or otherwise they have not been captured during the assay. Right? Next. Now what we are going to do with, as that we have seen our data now, we'll be moving further with some quality checking and selecting the cells. Which cells we'll be selecting, okay, for our further downstream analysis. So here you should know one thing is that we also have genes when it comes to animals and plants. Mitochondria also have its own genome, and in plants even chloroplast has its own genome, since this is a human PBMC data. So here we have mitochondria, and some genomic content of mitochondria will also be present. So what we are doing over here, that in our Surat object PBMC, we are creating a new column. So these, double brackets, double square brackets, what it says that in this particular object, one more column we are adding, and we'll be naming that column as %.mt, which will be storing the percentage of mitochondrial genome. Okay, and the function that we are calling is percentage feature set. Okay, for our PBMC Surat object. And what pattern we are searching? We are searching for the mitochondrial genome expression. So when I'm running this one here, so it has been run, right? Now for visualizing this, so number of, okay, different types of genes, different types of genes, and the total molecules, that how many RNA molecules are present, that you don't need to calculate. We have to only calculate the mitochondrial expression pattern, that's it. Other all things, that means the unique genes and the total number of UMIs, are already calculated when we had created our Surat object. You can find them stored in the object metadata. Okay, so let us see what are the metrics, quality checking metrics that are present, and for that we are only taking the first 5 cells. We can change this number, right? So if I run this, you see over here only the first 1, 2, 3, 4, 5 cells have been taken. This is the identity, and we have given the identity as PBMC3K. So this is the first column that is giving us the identity. No, this is the second column actually. The first column is giving us the BC value, that is the barcode value, and these represent different cells. So different cells will have different barcode sequences. Now N count RNA, that is the number of RNA molecules that are present in that for this particular cell, and N feature RNA, that feature means the number of genes that are being expressed. And what is the percentage of mitochondrial genomic expression? Is this 3.01, nearly that much expression of mitochondrial gene. So we have added one more metadata in our Surat object, right? So we can proceed like this. Next step will be visualizing our data. Let us also visualize it. We'll perform some visualization step. For visualization, we will be using a violin plot. To know what is a violin plot, you must know first what a box plot is. If you don't know what is a box plot, I'll be uploading a video, maybe in a short form content or a long form video, but not a big video. But I'll definitely try to upload that video, and I'll put the description of that video in the description box. Okay, link of the video, sorry, in the description box. And first, you should know what is a box plot, what does it signify. You should know what is a kernel density function, and then you will come to know what a Boylan plot is. But general explanation I'll give right now. First, let us run this. So there's a plot that has been generated over here. Let us maximize this plot. Yeah. So this is our data. For this data over here, for this graph, how to interpret is the first one is N feature RNA. So feature is nothing but the genes. Okay, N count is their expression value, and percentage empty is the percentage of the mitochondrial transcripts that have been one mitochondrial genome that is present, right? So x-axis refers to the spread of genome, the different features, the different genes. Uh, Overall, what you can see over here is the maximum value of the data point. See, the maximum data point is concentrated over here. Okay, so here N feature RNA that is telling you the number of unique genes detected per cell, right? And the second one, if you see the N count RNA, it is giving you the total UMI count per cell. Okay, that means if total UMI count is present, that means it is going to give you the total number of RNA content in a single cell. And percentage of mitochondrial gene expression is used for the how much mitochondrial genome is present, right? So that's a straightforward thing. So how do we interpret? So if you see the first one, N feature RNA, so cells with low values likely contain insufficient RNA. So the each point that you're looking over here, each dot that you're looking here is a single cell, right? So most of the cell here are concentrated over here. So what should be the number? Below 1,000. So most of the cells have genes below 1,000. Some of the cells are expressed. Some of the cells have genes that are expressed. Number of genes that are expressed are between 1,000 to 2,000. But we also get outliers, especially these 2 points. If you see, if I can zoom, no, I'm not able to zoom it. Okay, but here you see these 2 points over here, right? So these 2 points are the outlier. They are giving extremely high number of RNA, even more than the number of, sorry, high number of genes that are being expressed, more features. So they can be doublets. That means in a single gem bead, uh, two cells have been captured. So like that, we can detect outliers also. Okay. And when it comes to N count RNA, it correlates with the N feature RNA things. That means if you have how it correlates, it means that now again each point is the single cell. If you have abnormally high counts, um, that means number of RNA molecule is high. This also suggests that they may be doublets. So you see here also there are 2 points lying. So similar pattern we can see. So they both are positive. So number of genes are higher. That means the number of RNA molecules are also higher. Now what happened here is why do we look for percentage mitochondrial gene expression is the cells which are nearing their death or the cells which are already dead or cells which are not viable or which are under stress, they will express mitochondrial gene. Okay, so higher percentage of empty genes, mitochondrial genes suggest that they are not viable cell, their health is poor, right? So it's a good data that most of the points are concentrated between 0 to 5 percentage. So that's a good data. So that's it for this video, because the video is getting longer. I'll meet you in the next part. We'll continue with this script itself, right? So if any doubt, please feel free to comment down below, or you can message me in my LinkedIn, or in the WhatsApp group, or in the Google Classroom. I'll be there for the help. Thank you and have a nice day.